welcome to our webinar today on bioaerosol characterization, the air microbiome and Covid-19 transmission. So, good morning in Europe and good evening in Singapore. I'm Mark Richardson, I'm the CEO of the National Biofilm Innovation Centre here in the UK. And we aim to harness the UK's industrial and academic strengths in biofilms. And that includes collaborating internationally with our colleagues in Singapore. And this event is being held jointly with the Singapore National Biofilm Centre and SCELSE and supported by the Singapore Manufacturing Federation. And I'd just like to hand over to welcome you, Michelle Birnbaum, who's the SNBC lead at SCELSE. Michelle, over to you to say hello.
Thank you, Mark. Thank you very much, Mark. So I'll just give you a quick Reader's Digest version of both SCELSE and the Singapore National Biofilm Consortium because I want to leave room for the speakers. So SCELSE is the Center of Excellence for Microbiome and Biofilm that was set up in Singapore by the National Research Foundation. And it's about over 200 people that conduct research in both environmental and life sciences on biofilms and the microbiome. And SNBC, which is the Singapore National Biofilm Consortium, was set up to foster the ecosystem in Singapore and as well as internationally. And hence we have a very strong relationship with NBIC and also another one of our partners, which is CBE, the Center for Biofilm Engineering that's based in the US. And we're very happy to conduct those webinars. This is our third webinar, right, Mark? I think the third big international webinar.
Yeah.
And we were always lucky to have some very, very interesting speakers, and the same is true today. So I'll pass it back to you, Mark, so you can introduce our very first speaker.
So thank you very much, Michelle. So we've got a really interesting agenda for you this morning, as I'm sure you've already seen. And the first part of this session is going to be on the topic area of bioaerosol characterization. And I'm pleased to be able to introduce, to open that, Dr. Robert Ferguson, who's a lecturer from the School of Life Sciences at the University of Essex and also a member of BioAirNet, and which he'll explain what that is. And he's going to talk to us today about the characterization of bioaerosols. So, Robert, over to you to share with us your presentation.
Thanks. I'll just get the slides up for everyone. So yeah, hopefully you can all see that. If someone can give me a thumbs up. Good.
Yes, we can.
So yeah, I'm aware there's not much time. It's a short slot, so I'll get straight on into the talk. So I'm representing BioAirNet. I work at the University of Essex, and BioAirNet also involves people from Cranfield and some other institutions. And I'm going to talk about characterization of bioaerosols. So just a bit of background. There was a project before BioAirNet called RAMBI, which was a collaboration between Cranfield University and the University of Essex to look at bioaerosols across the UK. And this brought together researchers from Cranfield and Essex. So Fred Coulon, he was the PI at Cranfield, and then Professor Ian Colbeck, Dr. Corin Whitby, and now Professor Alex Dunbell at the University of Essex, and I was a postdoc on that grant. So what this research was really looking to take advantage was advances in analytical methods. So bioaerosols research has been significantly enhanced by these advances in analytical chemistry and molecular tools. And this is allowing faster, more sensitive and highly detailed characterization of air samples. And also I should add high throughput. So Cranfield and Fred and his team at Cranfield, they were looking at using chemical methods, so looking at phospholipids body acid analysis, so looking in detail at characterizing cell components, but also looking at trace gases released by the microbes, so volatile organic compounds. And then over at Essex, I'm a molecular ecologist mainly, so I was looking at extracting DNA and using methods such as qPCR for quantifying who and what's there and how much there is, and using high throughput sequencing to look at, well, who is there, but also potentially what they can do. Now, another advance in analytical methods that we shouldn't skip over as well is advances in statistical and computational tools, which allow us to actually make use of this data in a much more useful way. So what about BioAirNet, which is sort of the next step from this? Well, the goal of BioAirNet is to translate these tools, and also generally with our research, is to see how these tools can be taken out of the lab and put into the hands of end users, so their applications and their use by other people in a more practical situation. So really looking at how we can translate these advances in the characterization and quantification of microbiologically active components of BioPM, bioaerosols into technological solutions and process enhancements for end users. So, how can I get my, my molecular methods? How can we take them out to the sewage plant or the compost site? So the first thing we did in RAMBI is looking at the methods. So so some more specific stuff on what happened in RAMBI. So there's Ian and me with some basic air samplers using basic vacuum pumps right at the start of the RAMBI project. And what we were really thinking about then is there's lots of differences in sample collection. It's not really clear how as samplers. There's lots of differences in cell and DNA extraction efficiencies. And extraction efficiency also refers to the, is also a problem in the methods Fred's using over at Cranfield University with his chemicals analysis. And also, what are we looking for? We don't know what to look for because the biomarkers are poorly identified and quantified. So we need to work those out. But really, one of the overarching problems is when we're working with bioaerosols, typically, unless you go somewhere like a compost site, we're dealing with low biomass. And this makes it hard to apply these modern molecular methods, such as those that I use to air samples. And this is leaving our understanding of what's going on in the air lagging behind soil and water. And what this all leads to is, as a result, it's often difficult to link specific BioPM components to diseases, areas of exposure levels, and human health risks. Oh, sorry, just lost control of the slides. I've got a cat bursting into the room behind me. Sorry about that. So the thing we did is we set out to optimize air sampling methods. And this is a bit of a spoiler, but on this slide, what we discovered really is that sampling strategies, there's no good one size fits all rule. So it's more about fitting the choice of the sampling strategy to the question you need to address. So what we did is we looked at some different methods. So we looked at air filtration, we looked at liquid impingement methods, and we tried these with lots of different types of filters, lots of liquid matrices. We tried lots of different times, and we designed guidelines, user-friendly guidelines and recommendations for how to use these to collect bioaerosol samples to use with molecular methods. And if you want to read about that in detail, we have a paper. I can put the link in the chat after the talk, but it's in Molecular Ecology Resources, and you can go and have a look at that. But I'm just going to briefly cover the results. I won't do it in lots of detail because we'll run out of time. But one of the sort of big things we found is that two different methods, impingement, so bioaerosols being collected in a liquid, versus just simply filtering, that the filters recovered about an order of magnitude more 16S RNA copies than liquid impingement. However, both methods are just significantly correlated. So you can use both methods, but you're going to get more material with the filtration methods. However, it's not as simple as that. When we looked at the alpha diversity, so the number of different species we found in each sample, we actually found, although we're getting different amount, less material with the liquid impingement, we were getting much less diversity. So we were getting much less different types of bacteria. So the thing you're just looking at here is just that all of these lines are sort of sloping upwards with a blue box plot, much deeper description of the diversity present. And this is quite an interesting result and also quite counterintuitive. Why would we see this if that we're collecting less material? So one thing we started thinking about is basically literally what are we doing when we collect a bioaerosol sample? What's happening in time? And we know that bioaerosols and generally air pollution, it's not some static amount of thing like a bit of soil and you can just go and collect more of it to get more stuff. What you're doing is you're integrating over time. And you have to consider that when you collect bioaerosol samples. You're collecting these little snapshots of a very variable concentration of bioaerosols. And in its most simplest sense of diversity. However, the more you think about this, the more complicated it starts to get. Because with these short methods, the liquid impingement, what do they do? Well, they're collecting at much higher rates. So you're collecting a lot more material in a much shorter amount of time. The longer methods, they may be just skimming off the dominant species over a long time. So this sort of leaves us a bit of a problem. You start to think like, is it really possible to compare two methods just by normalizing for different amounts of time by normalizing the amount of volume of air collected, which is often what's done across the literature. And I don't have the answer to that, to whether it's possible, but it seems to me that there's a problem here. If we're going to collect lots of material in a short amount of time, that is not the same as collecting lots of material over a long amount of time. One thing gets you a lot of detail, a very detailed short snapshot. One of them gets a nice background look at what's dominantly there over a long period. But really what I want to summarize is there's no best method. We looked mainly at impingement and air filtration. They all have pros and they all have cons. So you have shorts capturing more diversity. The air filtration, we get lots of material and we smooth out noise. Now, what you can notice here is that the pros of impingement and the pros of air filtration, they're essentially a mirror image of each other. So that, as I said, you've got to match your method with your question. Do you need to get all the diversity? Do you need these shit over fine detail over long periods? Or do you want to know the background levels? Do you need to sample for a long time? So going forward, that's just something we need to think about as bioaerosols researchers. Now, another thing we've done, and this is a paper we recently published in Environment International, and something that we didn't look at in our previous work, and something that's maybe missing a lot, particularly in the molecular work, size matters with bioaerosols. The size of the particle matters to how far it will disperse, but it also matters to how deep it will be inhaled. So it's really important to understand, do bioaerosols, do the bacteria and the other components within them vary for different size classes? We went and did this in a compost site, and we found quite clear relationships between different bacterial groups and size components, both affecting their dispersal and their inhalation risk. And this is something that going forward, we really need to account for in dispersion modeling and public health risk assessment. So if all this talk about sampling methods and really detailed characterization and optimization has put you to sleep and it's not your thing, this is the point, I think, where you need to sort of check back in, and you might find this a bit more interesting and looking at some of the things that are what's next. And one of the things we've been working with is loop-mediated LAMP. Looking at this, well, we've been looking at low-cost sensing technologies, ways we can use our methods to sense specific pathogens in very short windows in the field. So we developed a simple assay that works by a color change that can be used in the field to detect key pathogens to act as an early warning system. So I'm just going to describe in detail just sort of overall what the tool does. So we take a environmental sample here. This is a Coriolis sampler. We recover those bioaerosols. So the main thing is we need to go pretty much with all bioaerosol sampling. We have our collective bioaerosols in a large volume, in this case 15 mils. We want to get as much of that as possible into a small molecular reaction. So we recover those bioaerosols. We get basically all of them into a tube. We set up a lamp reaction, and then that can either go on a heat block set at a temperature of 65 degrees for about half an hour, or it can go on one of these like Optigenie machines to do some quantitative lamp. And then within 30 to 60 minutes, you will know if the specific pathogen you're looking for was in the air. We've tested on this on Aspergillus fumigatus, obviously an obvious one to go for, but as well as other things such as Mycobacterium and Legionella. And that's a tool we're still looking to develop and maybe automate, things such as that. Now, another tool we've been working with on RAMBI is the SIB spectral intensity bioaerosol sensor, and I think you might hear more about similar tools later today. So it's certainly in terms of bioaerosols, this is one of the most advanced real-time methods. And it can tell you what tell you how many bioaerosols are in the air, what their size are. It looks at things on a particle by particle process, but it also generates lots of data on biofluorescence, allowing you to potentially discriminate between different types of bioaerosols. So this is something we've been trying to integrate with our molecular data. But looking forward, like this is a great system, provides fantastic amounts of data, really great detailed information, but it's not particularly portable. And this is something I've been thinking about is what can we do? Can we get a personal simplified version of this? We do lose a lot of data, but we have all these tools, apps, things people can carry around with them. They've been getting quite popular during the current coronavirus pandemic, little CO2 sensors. But could we have something like this, but for bioaerosols, something that could provide a little thing on your app? Yes, we wouldn't have the detail of the SIBs, but is there a way forward here? This would be fantastic for the field, right? We'd be able to collect detailed data on a wide level. So take-home message and future needs. So what information do we need that we don't have? Well, differentiation of sources. And we need sampling and detection systems capable of differentiating particle size and taking data which is spatiotemporally integrated, so measurements in real time. Which method should you use for your bioaerosol sampling? Well, it's going to depend on the question you ask. There's not one size fits all, one best method. You need to think about what you need to know and match the method. Maybe you need a combination of methods. Something we haven't talked about today, passive samplers, and something I've alluded to, small personal samplers. Can we bring that to the bioaerosols world? And also just opportunities and what comes next. Want to be involved in the network, get in contact with BioAirNet, so Fred Coulon at Cranfield, but also just to plug my similarly named BioSkyNet. I apologize, I gave it a similar name to Fred gave his. We've got a workshop coming in late July. Hopefully will lead to a nice global sampling project. It's late July, 4 workshops short workshops of about 2 to 3 hours. Get in contact with me. You can find me on Twitter at RMW Ferguson or my email rmw@essex.ac.uk. We're looking for members around the world. Come and work with us, come and do some research. And thank you for listening. And there's some email and contacts.
That's great. Thanks very much, Rob. So we'll take a few questions we've got coming in from the chat and a few other places. So the first one was, how do filter or impingement collection methods compare in relation to survival and viability of microbes when compared to molecular analysis?
So using them for culture-based analysis, yes? Is that the question? Yeah. So filtration, especially as you need such a long period, I wouldn't really recommend it for culture-based analysis because you're going to end up desiccating your samples, particularly if you're doing long periods. I know there can be a similar problem with impaction methods as well. I would say that impingement, if you're impinging into a liquid, that's probably your best bet for culture-based analysis. I note that one of the reasons impingement, one of the changes we made to impingement was impinging into PBS because it doesn't lyse the cells, basically, particularly when we freeze them. So I would argue that, yeah, that is a pretty decent way to go. It's also quite practical if you're going to culture out of them.
Great, thank you. So we've got quite a lot of questions here. Any that we don't have time to get to will be answered offline and sent round. So the next one is, did you get a chance to test a space that was not clean and then apply a cleaning product to understand what effect this has on the microbes identified?
In the air or?
So that came, I can't give you a name, an anonymous attendee asked that question. So if you could clarify that in the chat.
So actually the research I've been doing, we've been working outdoors. So I probably should have made that more clear. Like the work on Ramby was based, so we were sampling in places like compost sites, sewage sites, farm sites as some of our sort of dirtier, lots of bioaerosol sites, and then also some like urban rural parks, things like that, as sort of just some background. So we weren't working indoors with, an example to clean something. We do have a guy at Essex who is working on some sort of disinfection robots and is interested in that sort of thing, though. Seeing, sort of robots that would go into spaces and disinfect them. And he's wanting to know what will happen before and after when those robots operate disinfecting spaces.
Oh, very interesting. Have we got time for one more?
No, I think we need to move on.
Fine, yeah.
Thank you, Rob.
Sorry about my cat bursting in.
Okay.
I don't know how visible he was, but he just jumped on my lap and it's sort of, sorry about that.
Threw you off a bit.
Yeah.
Robert, so
I thought that was part of the presentation.
He's now in a box behind me.
Thank you.
I'll deal with some questions in the chat. Thank you, guys. Thank you for inviting me.
Thank you. So just a reminder, please, if you have any questions, can you pose them in the Q&A section like this? They're nicely segregated, and then William can go through them and kind of prioritize them. So for our next speaker, I'd like to introduce Dr. Jiayu Li, who's a postdoctoral scholar at the Berkeley Education Alliance for Research in Singapore, otherwise known as BEARS. And what Dr. Li works on is he focuses on the impact of indoor airflow patterns on indoor air quality, indoor and outdoor bioaerosol dynamics, and the sources of bioaerosol in the built environment. So the title of his talk today will be Study on the Size-Result Relationship between indoor and outdoor fluorescent biological aerosols particles by a real-time wideband integrated bioaerosol sensors. Mouthful. Here we go. Go ahead, Dr. Li.
Thanks for the nice introduction. I will share the screen. So can you see that well? Okay.
Yes.
Okay, sure. I will start. Okay. Thanks for inviting me to speak at the webinar. So good morning and good afternoon, everyone. My name is Jiayu Li, a postdoc researcher at Berkeley Education Alliance for Research in Singapore. BEARS is an overseas research institute of UC Berkeley and highly collaborate with local universities such as NTU and NUS. I'd like to use the next 15 minutes to share a recent case study in a bedroom within a larger project with the aim of understanding the relationship between indoor and outdoor bioaerosols in Singapore. So we choose to start with bedroom because it is the room which people spend most of their time in. We spend around, we spend around 65% of our time at homes. More specifically, 1/3 of our entire lives is spent in just one room, which is our bedroom. So the environment of our bedroom affects our health, well-being, and even your productivity next day. The common living conditions in Singapore is high-rise HDB public housing, which 81% of the local people are living inside. These residential buildings are commonly featured as under a window-based natural ventilation without air filtration system. So we focus on residential buildings not only because people stay longer in these places, but also because compared to commercial buildings or office buildings, occupants in these buildings are under relatively unfavorable scenarios regarding facing the pollutant from outdoors. So in this study, we focus on aerosols and bioaerosols. We want to know their relationship indoors and outdoors. What is the air? What is in the air which we breathe during 1/3 of our entire life, and how to mitigate the exposure? So with these questions, we started a 1-month bioaerosol measurement in a typical bedroom in Singapore, which is on a 6th floor of a total 12-floor building. During the 1-month experiment, one male researcher is using the bedroom as usual living condition. The main instrument for bioaerosol monitoring are two wideband integrated bioaerosol sensors, which I will introduce it in detail later. The indoor sampling point was on the table, and the outdoor one was 50 centimeters away from the wall under the window. So WIPS as a relatively new instrument for detecting bioaerosol, the principle is similar to the SIBS, also mentioned by Robert just now, and I would like to spend a little bit more time on this method. So WIPS featured as a single particle real-time instrument. From the side view, we can see that the aerosol particle are first drawn through the continuous wave laser, and the resulting scatter light is detected and used to count and size all the incoming particles. The scatter signal sequentially trigger 2 flash lamps filtered to emit UV light at 280 nanometer and 370 nanometer. Any fluorescent emitted by the particle due to these excitations is imaged onto 2 photomultiplier tubes equipped also with filters. One is from 310 to 400 nanometer, and the other is from 410 to 650 nanometer. So with the combination of these two excitation wavelengths and two detection range, and we can have three channels, three pieces of fluorescent information for each particles. And with the further combination of these three channels, we can further classify each particle into 7 fluorescent types. So, for example, if the particle only detected fluorescent at channel 1, but not show fluorescent at other two channels, we will classify it as type A. And if the particle detected in both channel 1 and channel 2, the particle will be type AB. And if the particle detected in all the three channels, and the particle will be type ABC. So with the width, we detected nearly 50 million particles indoor, and nearly 100 million those outdoors. And vast majority of them are non-fluorescent particles. And further, we calculate the concentration, the number concentration. We can see that the PM, the airborne PM concentration were 3.7 and 7.8 particles per cubic centimeter. And roughly 1/4 of them are fluorescent biological airborne particles. So in that bedroom, every fingernail size of air has a biological particle. So if we break down the concentrations of different type of particle into different sizes, we can see that the concentration of both fluorescent and non-fluorescent particle decreased with the increase of particle size. So if so there are much more smaller particles than larger ones. And if we calculate the particle concentration of, if we calculate the particle composition of PM in each size bin, we can see that in general, larger particles are more likely to be bioaerosols. And with the increase of particle size, the indoor fluorescent proportion in PM went from around 10% to nearly 100%. And if we compare between the indoor and outdoor PM composition, we can see that the proportion of fluorescent particles indoors are always higher than the proportion of that outdoors for each size bin. So if indoor aerosols are not solely from outdoors, and the indoor source generate more fluorescent particles than outdoors. And when we put indoor and outdoor information together, we can see that when the room was empty, the indoor upon outdoor ratio of both non-FBAP and FBAP decreased monotonically along with the particle size, which followed the rule of particle infiltration, which means large particles are normally harder to penetrate into indoors. If we open the window, and the ratio will be higher due to a higher infiltration rate. And more interesting findings was from the period when the room was occupied. We can see that, well, the ratio of non-FBAP still decreased as previous empty condition. And those of FBAP, the biological ones, first decreased at the first several bins, but dramatically increased at the size range of larger particles. And if we open the window, we can see that the ratio for the smaller particles were increased, but that for the large particle will drop. This indicates that outdoor is the major source of smaller particles, but larger particles are from indoors. So with the real-time indoor and outdoor aerosol concentrations, we can further quantify the indoor emission of particles with dynamic mass balance modeling. And the result shows that indoor emission rates were much higher when during the period when the occupant was in the room. Especially, one of the most interesting finding is that people still emit lots of particles when they are sleeping. Even it is around half of that when people awake. And the indoor and outdoor bioaerosols can be originated from both indoor and outdoors. With indoor source strengths, we also calculate the contribution of indoor source and outdoor source to indoor aerosol concentrations. It shows that indoor FBAP are more from indoor source, while the non-FBAP are more from outdoors. So at the last, with the indoor bioaerosol concentration, it's determined by the outdoor air quality and indoor activities. So we are thinking if we can use the station-reported air quality as an indicator to decide whether we should open the window or not. So we made several assumptions and also some simplifications. So we got two thresholds for awake and the sleep conditions. So if we use the air quality, the data, the station-reported air quality data in 2019, it's like nearly 19% of time is below the threshold for the awake condition. So our general recommendation is that opening the window when awake and close it during the sleep can lower the indoor bioaerosol exposure. Okay, so I will end my talk with some takeaway message from this study. So thanks for your attention. Please let me know if you have any comments and questions. Thank you.
Thank you, Jiayu.
Great, thanks very much. So we haven't got any questions in the Q&A box or the chat as of yet for your talk. If anyone wants to add any at any point in this presentation over the course of this webinar, please feel free to add them and we can get them answered offline and circulated to all the other attendees.
Okay.
Okay, thanks, Will. In which case I will move on to the next phase. We've had two very fascinating talks about the characterization of bioaerosols from Robert and from Jiayu and different measurement techniques. And the second phase of today's webinar will be very much considering the public health perspective of some of these findings that we're hearing about this morning. And to kick that off, I'd like to introduce Dr. Emma-Jane Goode, who's a research scientist from Public Health England. And Emma's going to talk to us today about the role that Public Health England plays in air microbiome research. So Emma-Jane, welcome this morning and over to you to give your presentation.
Thank you very much. I'm just share my screen. Hopefully everyone will be able to see it. Okay, so hello everyone. My name is Emma Goode. As mentioned, I work at Public Health England as a research scientist, and I wanted to talk to you today about what we do at Public Health England and the role we play in bioaerosols research and why we are interested. Now, normally I would put in a slide at this point to tell you a little bit about Public Health England, but we're currently undergoing a transition phase where we're being split up and moved into new organisations. So Public Health England will actually cease to exist in 6 months' time, and we will become the UK Health Security Agency. So if you stop seeing Public Health England being mentioned, we are still around, we just have a different name and a new brand. So bioaerosols, what are we interested in? So I'm sure you all know what a bioaerosol is, and we have a large number of groups working in Public Health England, and we're all interested in different aspects. So with the current COVID pandemic that we've all been dealing with, we have some very skilled research scientists at our Porton Down facilities who are looking at viruses and aerosolization of viruses and bacteria, and they do a lot of the work around infection and infection from bioaerosols, and including things such as Legionella research as well. Now, I work in the toxicology department, and we're actually more interested in fungi, and we're also interested in things such as pollen, but we're not so much interested in the infection side of things, because there are other health effects that we can get from our exposure to bioaerosols, and we focus more on these long-term health effects outside of infectious disease, and that is what our particular, or at least my particular interest is in. So as has been previously mentioned about particle size, it plays quite an important impact in public health. And so we do look at particles of all different sizes. We also have within Public Health England a nanotoxicology group. So they look at the very smallest of particles. They are the respirable particles, and they're the ones that we can be quite concerned with because they can accumulate in our lungs. We are interested in the different types of bioaerosols that we can get. So as mentioned in the previous presentation, we have a lot of indoor bioaerosols. As Robert mentioned, he's been looking at outdoor bioaerosols, and we are interested in all of these and how they can impact health, because we recognize very much so that people are exposed to different aerosols throughout the day. So they will wake up in their homes and they will be exposed to particular bioaerosols. They will go outside, they will travel to work, they will be at work, they will have recreational activities, and they're going to be exposed to a multitude of bioaerosols with varying content, and that is going to affect their overall health. So we are very much looking at, it's certainly in my group, the allergy, inflammation and sensitization health effects that we get, rather than just infection. Although infection is very important, and we've seen that more so than anything else in the last year, there are a range of other health effects that happen from bioaerosols. And allergy, inflammation and sensitization can cause quite a big impact on people's quality of life. So it's really, and can have quite a burden on the public health and national health systems. So it's really important that we understand how bioaerosols produce these health effects so that we can do the best we can to minimize the risk from these bioaerosols. Often with bioaerosols, the symptoms that we see are mostly respiratory in nature. So we can see that they include things like coughing, wheezing, shortness of breath, all things that can impact our day-to-day lives and prevent us from being able to do things that we would normally be otherwise able to do. But they can have non-respiratory symptoms as well, and lots of symptoms have been reported, such as gastrointestinal distress, fatigue, headache, and then long-term effects just on our mental health and well-being include things like depression, just all because it has such an impact on our lives. We see more often that those with pre-existing respiratory conditions such as asthma or chronic obstructive pulmonary disease, cystic fibrosis, can be far more prone to experiencing symptoms from exposure to bioaerosols, not least in terms of infection, but also allergy and inflammation. But we are also seeing now that there are a lot of other at-risk groups including those who are immune compromised. So anyone who's going through chemotherapy or has disorders or diseases such as HIV or chronic granulomatous disease, they are becoming more prone to health effects from bioaerosols. The most predominant one is infection, and we're starting to see a rise now in, and we have been seeing a rise for a while in invasive aspergillosis, as more and more people are using medications to dampen their immune system. So things like organ transplant as well is allowing our immune systems to stop being able to fight some of these the compounds in bioaerosols. On top of that, we also see people who work in places where they are exposed to bioaerosols on an occupational level. So farm workers, workers at composting facilities, and other industries as well, where we're seeing certain levels of particles in the air that can then link to adverse chronic respiratory health effects. And hypersensitivity pneumonitis is one such disease that we see a lot. On top of that, taking away occupational exposure and those with pre-existing health conditions, we are seeing sensitization in the population in general, certainly to, we see it a lot for people who don't have any pre-existing condition but do still suffer from things such as hay fever, will also can get sensitization to things such as fungi and fungal products that are also in the air. So it's not always the infectious side of things in terms of culturable infectious organisms. We get parts of organisms too that can have a real impact. And there's a lot of research being done at the moment on fungal proteases and our inhalation of those, and that causing sensitization and inflammation. So people can become sensitized over time, especially if they're being exposed on a daily basis in things like damp houses. We'll start seeing people become sensitized and then end up having an impact on their quality of life. So what do we do in Public Health England, and how do we work to minimise the risk? So there are 4 aspects of this, and there are sort of 3 ones that then lead on to the 4th. So we characterise exposures, we analyse health data, we understand the mechanisms behind it all, and then we use all of this to inform our decisions and our guidance and our interventions. So we're just going to give a little bit of an overview of each one. So I'm not going to go into big detail about characterising exposure because you've already heard quite a bit in detail from Robert, but we want to know, and it isn't necessary that Public Health England do the research. We do some research, we do some sampling, we do some analysis of the composition of bioaerosols, but we look to other institutions as well to publish their research and their findings that we can then gather those in. But we want to know what is in the bioaerosol, what are people actually being exposed to. We need to know how it changes throughout the year, and how it might change geographically or environmentally. We want to know where people are being exposed. Are they being exposed indoor, or are they being exposed outdoor? How does it change between indoor and outdoor? So we kind of want to build a really big picture of what we are being exposed to as we go throughout our day and where we're being exposed to these things. We do in Public Health England, we're currently doing a sampling project actually, where we're looking at we're looking at how the bioaerosols change in different locations. So we're using an urban, semi-urban and rural location, and we're sampling over the space of a year to have a look to see how fungal spore peaks change, what their concentrations are like across these different locations. And we do that using, we don't generally use culture or microscopy because we have found that it doesn't really tell us everything that we need to know, but we are using more molecular techniques as were previously described by Robert, and we certainly use DNA sequencing to give us the best picture that we can have of what is in our bioaerosols. So once we have got that picture, we can then start using that to analyze the health data. So we have teams within Public Health England are real-time syndromic surveillance teams, and they have access to a lot of different health data, and they're able to pick out the patterns and tell us when we're seeing certain things happening. So often at certain times of the year, we'll start to see peaks in people experiencing asthma symptoms. We'll start to see peaks in hospital admissions from people with asthma attacks. And we can use that kind of data to understand or link them to certain things that we're seeing in our exposures in what our bioaerosols are showing us. So we know that we'll see seasonal variations in fungal spore concentrations, and we can start to link them to the health data, to the peaks that we're seeing in symptoms in the general population, or perhaps in a particular subset who are dealing with a particular pre-existing respiratory condition, or a particular cohort of people who, in terms of where they work, where they live, etc. So essentially the questions when we're analysing the health data is who is being exposed, who is experiencing the health outcomes, when are they being exposed, when are we seeing these health effects, when are the peaks and troughs in symptoms and things like that. And we want to know how they're being exposed. So are they being exposed outdoor, indoor, are they near to do they live near to a particular waste processing site, for example, or do they live in an urban area, those kind of things. And we're trying to identify those associations between the specific health outcomes that we're seeing and what we are seeing in the bioaerosols. And once we've got all of that, we understand that thing A in the bioaerosol is leading to outcome B, we can then start to look at understanding the mechanisms behind it. So getting into the cellular and molecular biology and understanding why exposure leads to a particular health effect. So what is the pathway? What is going wrong? What is causing this symptom? And finding those links between exposure and disease. And my particular focus at Public Health England is on understanding the mechanisms. And I'm currently doing some work looking at how the pathways is slightly different in terms of recognition of fungal spores in asthma and people who suffer with allergy. So once we have all these three things, we then use that to inform our decisions and our interventions. And I kind of liken this a lot to, if you've ever done your mandatory fire training, you'll have been taught about the fire triangle, where you've got heat and oxygen and fuel, and that creates fire. And if you take one of those things away, the fire goes out. And I feel the same comes with public health research and guidance. We need to characterize exposures, we need to analyze the health data, we need to understand the mechanisms in order for us to put out the best advice, recommendations, guidance and interventions as we possibly can. We need all of that to make sure that our recommendations are robust, based on the science, based on the evidence, in order to make sure that people's health can be protected as best as possible. So we don't do all this ourselves. We do rely on a lot of people, a lot of other institutions to do the research. We often review the research. We do large-scale literature reviews, systematic literature reviews, in order to understand what other people have found out, that we can bring it all together and help to protect the public. So we do this in many different ways. We will often put out advice. You'll see guidance documents. You'll have seen many over the last year coming out from COVID that we'll put out to say you should do this, you should do this, this is how you protect yourself, those kind of things. We also, in certain cases, will also start alert systems, early warning systems. We already have these kind of things for other non-biological things such as heat waves, cold weather, flooding, for example. But we want to now start looking at how we might be able to take the research around bioaerosols and use that to create better early warning systems for people with particular pre-existing respiratory conditions that might be affected. We can also make recommendations, and that's not just to the public. We can do that to local authorities, to central government, to different departments and organisations and businesses. So an example would be if a local authority contacted us because there's been a planning permission for for a housing estate to be put next to, say, for example, a waste or compost site, we could say, well, we would recommend that you don't have any houses within a set amount of distance, because you're likely to see an increase in adverse health effects from being close to that site. Or we can input into the regulations. So we work closely with other organisations such as the Environment Agency, where we can feed into their regulations that they then put out to make sure that, again, everybody stays safe. So we take all of that, hopefully produce the best advice and guidance that we can to protect health. But it never stops. It's like painting the Forth Road Bridge. We're constantly having to go back and analyse and read and have another look at all the evidence that's out there, all the research that's out there, so that we can continue to constantly update that guidance so that's always the best that it can be. And there's also things that come through. So we think we understand something, and then something changes, or we get these emerging issues that come through in bioaerosol research. And there is a few here that we're starting to look at now in Public Health England. And the first is around co-exposures. So we often can break things down into its simplistic form, and we look at our exposure to fungi, we might look at our exposure to viruses or our exposure to bacteria, but it is so much more complicated than that, because in the air we breathe are so many more particles that we take in. So if you live in a very urban area where we've got a lot of air pollution, you're also being exposed to that. That's having a health effect. And then you might be exposed to fungi, and that's going to have an even bigger effect. And we need to understand how exposure to all of this impacts and compounds the situation, rather than just looking at it in its own each individual form. We need to look at it in each individual form, but now we need to start bringing it together and understand how each one impacts one another. And then we're also starting to look now at environmental change. So climate change is a big thing on everyone's agenda, and we know that it's going to change, likely to change the biodiversity in across the world. So that's going to mean changes in bioaerosol composition and concentration. And we're also going to start seeing things such as intensive rain and flooding, and this is going to increase things like the prevalence of mold and dampness in housing. So this is going to again change the bioaerosol compositions that we see across the board. And then finally, we're starting to understand a little bit more that about the human microbiome, and I mean this in a couple of ways. So we all have started to learn, or we all know now, that the gut microbiome is able to influence our overall health, and we're starting to understand that there's now this gut-lung axis, and that the gut and the lung are able to talk to each other, especially as they are both systems that have exposure to external factors, and that is able to influence our overall health. So we want to understand a little bit more about that, but also we're starting to understand, previously I think everyone assumed that the lungs were quite sterile, but we're now starting to learn that that might not be the case, and that we might be seeing sort of more colonization of airways, especially in people with pre-existing respiratory conditions. So that is an emerging issue as well, which I hope other people are also looking into, so that we can use your research to help inform public health outcomes. So that is just a very brief overview of what we're doing in Public Health England and what we're looking at and why we're interested. So thank you very much for your attention. I just wanted to acknowledge BioAirNet, of which I'm also a member of, and the Health Protection Research Units, which are funded by the National Institute for Health Research, of which we do a lot of our research out of. So thank you very much for your time.
Great, thanks very much. Thanks very much, Emma. So one of the questions we've had come in is who sets the limits for some of the things you've spoken out, and do PHE influence changes in those limits?
Yeah, we will. It's not us to set the limits. If it's regulatory side, that will be the Environment Agency. There will be the one who will be responsible for that. But where is certainly conversations that happen, we will liaise with them. They'll ask as well about whether or not we see any health effects, and as we do our research, again, we'll feed into that as and when we get answers for their questions. But yes, it's a two-way thing. We don't set them, but we certainly have an input into them.
Sure, great, thanks very much. And if we have any other questions for any of the previous speakers, please do put them into the Q&A box and just mention the name of the speaker so I can make sure it gets to the right person and they'll get circulated after the webinar. Thanks very much.
So we'll move on to the next speakers from the Singapore Disinfection Task Force. So Ms. Francesca Solis and Ms. Amanda Yeo are both members of the COVID-19 Cleaning and Disinfectant Industry Task Force in Singapore, of which SNBC is a contributing technical member. So this task force was set up by the Singapore Manufacturing Federation's Energy and Chemical Industry Group, and it was set up to be a platform for the cleaning, disinfection, and hygiene industry. And currently, the members of this task force are companies such as Vance Chemical, ICMO, 3M, Supersteam, and P&G. And the title of so they're going to give us an overview of CDITF, which is the task force, and also the disinfectant industry guidance document that has been put together. Over to you, ladies.
Francia, I think you are on mute. Oh.
I think you're on mute, Francia.
Hi, can you hear me?
Now we can, yep.
Okay, sorry about that. So, thank you so much. We're very thrilled for this collaboration and with the organizers, and I'd like to wish everyone a very good day and good health. I am here to introduce to you briefly the Singapore COVID-19 Cleaning and Disinfection Industry Task Force, which I am grateful to be part of and have the opportunity to help steer the industry through COVID-19. COVID-19 came rather unexpectedly and has evolved rapidly. And along this is the surge and demand in cleaning products, as well as increase in businesses in disinfection industry. So with a number of scattered information, a lot of confusions regarding products, practices, and regulations on cleaning and disinfection were formed. This is why the Singapore Manufacturing Federation has launched this task force under the Energy and Chemicals Industry Group to have a centralized team to tackle some of the industry's concerns. I am honored to be working with such a dynamic members coming from local and multinational companies and academic associations, and having different backgrounds make it easier for us to address the industry concerns and help us meet our objectives, which we set right when we established the task force. The situation will continue to evolve, and our mission must evolve along with it while we remain focused on our objectives. First off is COVID info sharing. We aim to discuss issues that can affect the industry and public safety, including regulatory updates from local agencies, where we try to gather concerns from interested parties. We also want to promote innovation through capacity building of technical expertise in the industry. This is why we hold webinars and support webinars such as this, as we see it as an opportunity for the industry to gain more knowledge. So through collaboration with academic associations such as SNBC and SCELSE, and with some government agencies as well, we also strive to share and support the best practices to encourage the industry's competitiveness. Another important objective that we want to achieve is to encourage the manufacturers and the sellers to ensure quality and safe products by developing standards and guidelines, which later on will be shared to you. And of course, to educate the public about it. So whether you're from the industry or from the academe, please do reach out to us if you're at all interested to contribute or if you have any concerns. So to share further, may I introduce to you a fellow CDITF member who has significantly contributed to one of our initiatives, the Disinfectant Industry Guidelines. Ms. Amanda Yo, please.
Hi, so my name's Amanda. So basically, we actually came up with one of our initiatives, which was to develop a cleaning guideline, which was to develop a disinfectant guidance for the industry. Because the Singapore COVID task force, basically what we saw was that there was a gap in the industry because disinfectants are actually not regulated in Singapore. At this moment, at that moment, there was no specific guidance, there were no label requirements, and there was a lack of proper guidance and a lack of standardization. So what we saw that this would bring about for confusion for the consumers. And when we say consumers, we're not just talking about the man on the street, but even like maybe some of the smaller players and some of the manufacturers in the market. Next slide, please. So because of that, we actually developed this disinfectant guidance, and our purpose was to create an equal playing field. It's developed by industry for industry, and we actually sought for insight from NEA as well. And this industry is a benchmark for quality, safety, and efficacy of surface disinfectants. This is a voluntary guide, so it's not regulation or anything, but we thought it's a collection of all the best practices. So in case, industry players who did not know what to do, or they wanted to understand more or have more information, this could be a guidance for them. And the most important thing was that we wanted to foster appropriate use. Next slide, please. Okay, so basically, as we mentioned, the scope of it is actually, it only is hard, non-porous surface disinfectant, which can be used in concentrate or ready-to-use format. So we don't include other types of disinfectant, like maybe your cloth disinfectants or your laundry. We're only looking at the hard, non-porous surface disinfectants. And the primary function shall be to disinfect. So this is the main function. So we're not going to, so this does not cover products where like the main function is actually more of like a fragrance or some other purpose or cleaning. This is really a disinfectant. And we also just do not apply to any of the following products, like products which are regulated by health authority, such as cosmetic products, antiseptics, or skin sanitizers. We also don't cover water only, any products that is primarily used for general cleaning, and any freshener products that is primarily used for freshening the air, or products that's used on soft porous surfaces like fabric or leather. Yeah. Next slide, please. Okay, so the key points that we actually went through in our guidance document were like the scope and purpose, which we shared earlier, the different definitions that we had in our guidances, like what is antibacterial or what is antiviral. We talked a bit about formulation, like what are some of the good practices that you needed to have in order to have a good formulation, and how do we ensure efficacy of products? Because I think this is a very important thing. Like if we say that our product can kill 99.9% of bacteria, what does that actually mean? How do we actually need to do it? And we talked a little bit about stability, packaging information that we think would be useful and important for the consumers. And we also propose a standardized labeling. I think labeling is important because this is the information that your consumers see. So we thought that, if you can have a standardized labeling, at least the consumers can compare and they'll be able to make the best decision for their products. We also had claim substantiation information. How do you substantiate your claims? What documents and records do you actually need to have and keep, as well as references for all the other international website or international regulations and guidances that we were looking at when developing this guidance document. Next slide, please. Okay, so basically we have already released this disinfectant guidance for industry use under SMF. So if anyone is interested, please go to our SMF website. You'll be able to find our disinfectant guidance down there. We actually also conducted a webinar on the 23rd of February to a wider audience to explain our webinar and also to ask for any further comments or any further guidance that the industry is interested in. And of course, we'll be looking at other trainings depending on whether or not there's a need for this. So basically, yeah, this is one of what we wanted to share of some of the activities that we are doing. And if there's anybody who's interested to know more about SMF or about our task force, please feel free to reach out, and we would love to answer questions, and we would love to hear from the industry as well. What done with with just sharing about our task force and the disinfectant guidance. Thank you very much. Friend, do we have anything else to share? If not, yeah.
Great, thanks very much. Just got one quick question before we finish. Do the standards that you've set out in your talk, do they involve any biofilm testing at all?
Sorry, I couldn't hear that. Could you say a bit? Can you repeat the question a bit louder? Sorry.
Sure. Sorry. Yeah, my internet connection is not great. Just a quick question. The standards that you've set out in your presentation, do they involve any biofilm testing at all?
No, we didn't actually look at biofilm testing. So it was really looking more on like just the normal disinfectant. So we didn't actually have much information on biofilm.
Okay. Great. Thanks very much.
Okay. So thank you very much to all of our speakers today, and thank you very much everyone for attending. Just to let you know that any questions that have been unable to be answered today will be circulated post the meeting to those people who asked them. Most but not all of the presentations will be posted on our joint websites in a few days, and we do ask you to give us any feedback you may have about the meeting directly to either SNBC or National Biofilm Innovation Centre in due course. So, Michelle, is there anything you wanted to say finally before we close?
I just wanted to say thank you to all the panelists for excellent presentation and for their time, and all the attendees. I hope everybody enjoyed the presentation, and that each found something useful in those presentations. And as Mark said, if you have any feedback, good or bad, please let us know, because the intent is to keep improving those webinars so it serves the community. Thank you very much.
Thank you, and thank you. So thank you all very much for attending today. If there are further questions, do post them until we completely close the webinar in a few minutes' time, and we'll make sure those get answered. So thank you, for example, Yudi, for your question on detection methodologies. We will circulate that to our panelists and get that answered. So thank you all very much. Have a good evening and a good afternoon here in Europe, and a good evening in Singapore and the wider region. Thank you.
Thank you, everybody. Good night.